Hello everybody. In this session we are going to discuss about two topics. One is the transport of oxygen and carbon dioxide in the blood. Along with that we will be discussing chloride shift, Haldane effect, and then we will discuss what is this oxygen hemoglobin dissociation curve. First we will discuss the transport of oxygen and carbon dioxide in the blood. See, let us assume these are the body cells. These are the body cells. Okay, and during respiration you know that these cells release carbon dioxide into the intercellular spaces outside into the lymph. So the carbon dioxide comes into the lymph from the cells during respiration. Now where does this carbon dioxide go from lymph? It goes into the blood. Let us show one blood vessel. I'm purposely showing the blood vessel in large size just to see what's present inside the red blood cell. In the red blood cell, definitely you have in the blood vessel you have the red blood cells. So this is one red blood cell. So we're just showing it a bit large to see what's inside. Okay, and outside the red blood cell you have plasma. This is the plasma. So outside you have lymph and inside you have plasma. This carbon dioxide, 7% of this carbon dioxide present in the lymph, it goes and dissolves in the plasma. Okay, it's dissolved in plasma, and that the carbon dioxide which is dissolved in plasma will slowly react with the water, because in plasma you have 95% water. So it slowly reacts with that and forms carbonic acid. This is very slow carbonic acid that soon dissociates into H plus and HCO3 minus. And this H plus is the cause for the acidic property of impure blood. You know, the impure blood, the venous blood is more acidic than pure blood or the arterial blood. Why? Because in impure blood there is more carbon dioxide in the plasma, which is combining with the water forming carbonic acid, and then that's giving H plus ions. These H plus ions are more if there is more CO2, and that causes acidic property of the venous blood. And about 70% of this carbon dioxide, it goes and combines with the CO in the water in the red blood cell. Okay, that CO2, it combines with water. The same reaction, you get carbonic acid, and that will soon dissociate into H plus and HCO3 minus. The same reaction, but here it's very fast because you have one enzyme over here, that is carbonic anhydrase. Okay, so the carbon dioxide which is dissolved in plasma is slowly converting into carbonic acid and H plus and HCO3 minus. But the carbon dioxide which is going into the red blood cell will react with water and form carbonic acid, but it's a bit fast because of the enzyme carbonic anhydrase. And what about the remaining 3%? The remaining 3% of carbon dioxide, sorry, this is the carbon dioxide, and the remaining 3% of carbon dioxide, it combines with the hemoglobin in the red blood cell, Hb, forming HbCO2, which is carbaminohemoglobin. Carbaminohemoglobin. This carbon dioxide, it combines with hemoglobin and forms carbaminohemoglobin, just like how oxygen combines with hemoglobin. You know, one hemoglobin molecule has 4 hemes, so each heme can combine with an oxygen. So one hemoglobin can combine with 4 oxygen molecules, and the oxygen actually binds with the iron of the heme. But where does the carbon dioxide bind? The carbon dioxide binds with the amino terminal of the hemoglobin. See, for example, when you see the hemoglobin structure, it's like this. The hemoglobin has 4 polypeptide chains. This is the alpha 1 chain, alpha 2 chain, beta 1 and beta 2, and here is the heme. This is the heme. So the hemoglobin molecule has 4 chains, 4 polypeptide chains, alpha 1, alpha 2, beta 1, beta 2, and 4 hemes. So each heme can combine with an oxygen and forms a HbO2, oxyhemoglobin. In the same way, that's when in the alveoli. it combines with oxygen. But when this hemoglobin reaches the tissues where there is a lot of CO2, that CO2 it combines with the somewhere you have the amino terminal. There it combines CO2, CO2, CO2 and CO2. This is carbamide hemoglobin. So just remember, oxygen combines with the heme, but carbon dioxide combines with the amino terminal of each polypeptide chain. So just like how one hemoglobin can combine with 4 oxygen molecules, one hemoglobin molecule can also combine with 4 carbon dioxide molecules. So this is what you see here. So Hb plus CO2 gives HbCO2, which is carbamide hemoglobin, and that is 3%. This happens when the hemoglobin is near the tissues. But when the but look at here, this H plus ions formed inside the red blood cell will not the H plus, the bicarbonate ions. These bicarbonate ions which are formed inside the red blood cell will soon diffuse out of the red blood cell into the plasma. It will come here, HCO3 minus. Okay, the bicarbonates which are formed in the red blood cell will immediately diffuse from the red blood cell out of the red blood cell into the plasma. But when there are more and more bicarbonate ions coming out of the red blood cell, there is an ionic imbalance. So there is an danger in the increase in osmotic potential. So to counterbalance, to regulate this ionic balance, chloride ions move from the plasma into the red blood cell. So when more bicarbonates come out of the red blood cell, there is accumulation of more negative charge and there's an ionic imbalance. To counterbalance that, the chloride ions move from plasma into the Cl minus. But from where did you get this Cl minus? You always have right here. You always have NaCl in the plasma. This NaCl, it will dissociate into Na plus and Cl minus, and the NaCl will dissociate into Na plus, Cl minus, and that Cl minus will come into the red blood cell. And this Na plus and HCO3, they both combine to form NaHCO3, sodium bicarbonate in the plasma. This movement, this drift of chloride ions into the red blood cell is called as a chloride shift. That's called as chloride shift. Okay, so what's happening here? The carbon 70% of carbon dioxide which has entered into the red blood cell, it combines with the water and fastly gets converted to carbonic acid, and that fastly dissociates into H plus, HCO3 minus. The HCO3 minus come out of the red blood cell, and in return the chloride ions come into the red blood cell. This is called as chloride shift. Okay, that's the chloride shift. Now coming to now we will discuss one important curve, the oxyhemoglobin dissociation curve. What is oxyhemoglobin dissociation curve? It's a curve which shows, it's a graph which shows the relation between the partial pressure of oxygen in the blood versus the percentage of hemoglobin that is saturated with oxygen. So the curve, it looks like this. This is the x-axis, this is the y-axis, the origin, and let's divide this into 10. So here on x-axis we take the partial pressure of oxygen, and on y-axis we take the percentage of hemoglobin that is saturated with oxygen, percentage of hemoglobin that is saturated with oxygen, and we measure here the partial pressure of oxygen we measure in millimeter of mercury, also called as torr. So 10 torr, 20 torr, 30, 40, 50, 60, 70, 80, 90, 100, 110, like that. This is in millimeter of mercury, or it's also called as torr, because 1 mm of mercury is 1 torr. And similarly here, 10, 20, 30, 40, 50, 60, 70, 80, 90, 100, and thereupon. This is the percentage and this is the torr. See here. So this is the graph, and the graph, it looks like this. It slowly starts. First there is slow rise in the curve, and then there is a sudden steep rise, and then it becomes stationary at about 100%, because 100% meaning the complete hemoglobin has been saturated with oxygen, and there is no more rise. It becomes stationary. When it has reaches 100%, meaning all the hemoglobin has been saturated with oxygen and there is no more increase. It becomes stationary at 100%. Now, see here, the curve has slowly raised with increase in partial pressure of oxygen. It has slowly raised, slow rise. Why? That is because of the cooperativity of hemoglobin with oxygen. See, for example, if you look at hemoglobin, I don't want to show the whole structure, I'll just show you the rough structure of hemoglobin. This is the hemoglobin molecule, and it has four hemes. This is one heme, this, and this. Four hemes. When there is little oxygen in the blood, so there is little partial pressure of oxygen, so one oxygen molecule binds with the hemoglobin. But when one oxygen molecule combines with one hemoglobin, there is an increase of affinity for the other hemes with other oxygens. Okay, when one oxygen molecule binds with one heme, the other hemes are more, they are more desirous. They have more affinity for another oxygen molecule. So another oxygen molecule binds with another heme. So there is still increase in affinity. So one more oxygen binds more fastly with another heme, and there is still very high affinity. So another oxygen molecule binds another heme. So totally one hemoglobin has bound with four oxygens. But how does it happen? So when one oxygen molecule combines with one heme, there is an increase in affinity for the other hemes to combine with oxygen molecule. This is called as cooperativity. So initially when there is oxygen coming, the affinity slowly increases. The affinity for the oxygen by the hemoglobin increases. That's why there is a slow rise in the curve. This is called as cooperativity, or cooperative binding of oxygen with hemoglobin. So because of this reason, there is a slow rise in the curve. But as more and more oxygen is combined with hemoglobin, the affinity for hemoglobin also increases, and there is a steep rise, and then it becomes stationary. So this is the oxyhemoglobin dissociation curve. And this, so what is it? What is this? It is a graph which represents the relationship between partial pressure of oxygen in the blood with the percentage of hemoglobin that is saturated with oxygen in the blood. But this oxyhemoglobin dissociation curve is very dynamic. It's not in one place. It moves to the right side. It moves to the right side in tissues. It's like this. So this is in tissues. It moves to the right side. It moves to right in tissues, and then the same blood, when it goes to the alveoli where there is more oxygen, it shifts to the red, to the left side. This is it happens in alveoli, because in alveoli there is more oxygen, because the air you breathe in has 21% of oxygen and 0.3% of carbon dioxide. So definitely the air what you breathe in, it goes into the alveoli, and in the alveoli there is a high partial pressure of oxygen. So the curve shifts to the left, to the left, to the left. So this oxyhemoglobin dissociation curve is very unstable. It is very dynamic. It moves to the right side when the blood goes to tissues, and it moves to the left side when it goes to the alveoli. So why does it move to the right side when it goes to tissues? Why? Because in tissues there is more carbon dioxide released. In the tissues there is more carbon dioxide released. So there is a high partial pressure of oxygen. So that high partial pressure of oxygen, and definitely comparatively there will be low partial pressure of, so it's the high partial pressure of carbon dioxide, and comparatively there will be a low partial pressure of oxygen. And also the temperature is high compared to the alveoli. The temperature in the tissues, it's high, 37 degrees Celsius. So because our blood is it's warm, more heat is released. So in the tissues there is more temperature, and also increase in one compound called 2,3 diphosphoglyceric acid, that also increases. So all these factors, they cause the oxyhemoglobin dissociation curve to shift to the right side, which happens in the tissues. That is because of more carbon dioxide, less oxygen, more temperature, more 2,3 diphosphoglyceric acid. So like that, you have different factors which will cause the curve to move to the right side. And there is also um, increase in acidity also. Increase in acidity. But because I told you, in the tissues, when there is more CO2 coming here, there is more H plus ions. Okay, in the tissues, when there is more carbon dioxide which goes into the blood over there, it forms this H plus ions which is acidic. So that acidic increase in acidity of the blood also shifts the curve to the right side. So all these are the factors which make the oxyhemoglobin dissociation curve to shift to the right side, which happens in the tissues. And when the same blood is going to the alveoli, you have the reverse. You have the reverse. What happens? The carbon dioxide, it will in the alveoli it is less, and the oxygen is more, which is 21 percentage, and the temperature is comparatively less, and 2,3 diphosphoglyceric acid is also less, and the acidity is also less. So these factors are available in the alveoli which make the curve to shift to the left, and these factors are available in the tissues which makes the curve to shift to the right side. So when the curve is shifting to the left right side, what's happening actually? The carbon dioxide is being displaced. This is actually called as Bohr effect. This is called as Bohr effect. Bohr effect, where the hemoglobin is combining with CO2, and it's forming HbCO2. That's Bohr effect when it's shifting to the right side. But this one, when the curve is shifting to the left, that's called as Haldane effect. Haldane effect, where the Hb O2 will dissociate into Hb plus oxygen. Here the oxygen is displaced, but here, sorry, no, the oxygen, the hemoglobin combines with oxygen to form HbO2, and here actually what happens is the HbO2 dissociates into Hb plus oxygen. So in the tissues, when there is more oxygen, there is carbon dioxide is displaced and it combines with oxygen to form oxyhemoglobin. This is Haldane effect. But in the tissues, when there is when there is more carbon dioxide and less oxygen, what happens? The HbO2 will dissociate into Hb plus oxygen. That is called Bohr effect, and this is the Haldane effect. And the same thing, in the same observations, what you see in this oxyhemoglobin dissociation curve, we'll see in reality how does it appear. For example, let's think this is the alveolus. This is the alveolus, and in the alveolus there is more oxygen, I said. So there is more partial pressure of oxygen. It is 104 millimeters of mercury or 104 torr, and the carbon dioxide is less, that is only 40 torr. This is the standard value of partial pressure of oxygen in alveoli, 104 torr, and partial pressure of carbon dioxide is 40 torr. Now I'll show you the heart, the rough structure. This is the heart, and the four chambers. This is the right auricle, the left auricle, This is the right ventricle and left ventricle. And you know, the systemic veins, like the superior inferior vena cava, they bring the impure blood to the right auricle first. The impure blood comes to the right auricle, and from there, the impure blood goes to the tricuspid valve into right ventricle, into right ventricle. Okay, from right ventricle through the pulmonary artery, through the from right ventricle through the pulmonary artery, you have the impure blood coming out, coming out. So like this, the blood goes here. So this is the systemic veins which bring the impure blood to the heart, the deoxygenated blood, like superior and inferior vena cava, which comes to right auricle. From there, the tricuspid valve it goes down into the right ventricle, and from there the impure blood goes into the pulmonary artery. This is the pulmonary artery, and from there, the blood it goes to the alveoli. Purposely, I'm showing you here the blood was in a large diameter. I want to show what's there inside the uh, right artery. So one thing, remember, what is the partial pressure of oxygen in this deoxygenated blood? The partial pressure of oxygen in the deoxygenated blood is 40 torr, and the partial pressure of carbon dioxide is 45 torr. So what's happening here? The partial pressure of oxygen in the alveolus is 104, and here it is 40. So it's more here. So definitely the oxygen will diffuse from the alveolus into the pulmonary artery. The oxygen will diffuse here, and when the oxygen diffuses here, the partial pressure increases to something like it goes to 90, 97, 97. Sorry, it goes to 95, 95 torr. And here there is more carbon dioxide here and less carbon dioxide here. So what happens? The carbon dioxide will diffuse into the alveoli. And when carbon dioxide leaves the pulmonary artery, here the partial pressure of carbon dioxide will drop down. It will drop down to 40 torr. 40 torr. Now this blood, so I'll write this with red because that becomes pure blood now. This is 95 and this is 40. Okay, so what's happening here? Here the partial pressure of oxygen is increasing from 40 to 95, and the carbon dioxide partial pressure is decreasing from 45 to 40. This blood, it goes, it comes into the left auricle. It goes into the left auricle. From here I'll show with red because now that's pure blood. It goes here into the left auricle. The pure blood it comes into the left auricle, and that is actually called as the pulmonary vein. This is the pulmonary vein. So through the pulmonary vein, it goes into the left auricle, and here the blood is coming like this. So the deoxygenated blood which is coming from the pulmonary artery has these values. O2 is 40 and CO2 is 45, but that oxygen will increase to 95 and carbon dioxide will drop to 40. That's the oxygenated blood which comes out of the lungs through the pulmonary vein into the left auricle. From left auricle through the tricuspid valve, the pure blood it goes down into left ventricle, and from left ventricle again that pure blood it comes out of the left auricle through the systemic aorta. So like this. Now this is the pure blood. The pure blood or the oxygen blood it goes to the tissues. It goes to the tissues. Okay, now let's show these are the tissues. So these are the tissues. Now, what is the partial pressure of carbon dioxide in the tissues? The partial pressure of carbon dioxide in the tissues is high. It is 45 torr, and the partial pressure of oxygen is 40 torr. 40. So this same blood, which is having partial pressure 95 and 40, will go to the heart. From heart, it comes over here. Okay. So the same blood you have here. So again, I'll show you this. I'll show you in a bigger diameter. Okay. Now, what is the partial pressure of oxygen here? I told you the partial pressure of oxygen is 95 because it is that same blood which is coming to heart and from heart to the cells. And partial pressure of carbon dioxide, it is 40. Okay. Okay, I'll write carbon dioxide this side, which is 40, and I'll write the oxygen here, which is how much? 95. Okay. Now, see, the same blood, oxygen is 95, carbon dioxide is 40. So oxygen is 95, carbon dioxide is 40. Now, look at here. There is more partial pressure of carbon dioxide oxygen here and less partial pressure of oxygen is here. So what happens? The blood will diffuse from the blood capillaries to the tissues. The oxygen will diffuse. The oxygen will diffuse into the cells, and in return, the carbon dioxide, it has having a more partial pressure of in the tissues, so it diffuses into the blood capillaries. This is the carbon dioxide. So when that happens, what is happening? So there is more oxygen leaving the blood capillaries into the tissues. So again, the partial pressure of oxygen will drop down from 95 to, it will drop down from 95 to, it goes to 40. And the partial pressure of carbon dioxide, as more and more carbon dioxide is coming inside, it will increase from 40 to 45. And that same blood will go to the heart. From heart, it goes to here, into the alveoli. See, the oxygen is 40, oxygen is 40, carbon dioxide is 45, carbon dioxide is 45. The same blood, it goes here. So like that, you can see the deoxygenated blood going into the heart, from heart to the alveoli. Here, when the pure blood, 100 ml of 100 ml of oxygenated blood, 100 ml of oxygenated blood can deliver up to 5 ml of oxygen, 5 ml of oxygen to the tissues. But here, 100 ml of deoxygenated blood, 100 ml of 100 ml of deoxygenated blood, it can deliver up to only 4 ml of carbon dioxide to the tissues. So is that clear? So 100 ml of oxygenated blood can deliver 5 ml of oxygen to the tissues, and 100 ml of deoxygenated blood can deliver 4 ml of carbon dioxide to the alveoli. I want you all to observe one thing over here. In the pure blood, where the partial pressure of oxygen is 95, what is the hemoglobin percentage? You can find out now looking at this graph. When the partial pressure of oxygen is 95, somewhere here it's 95, what is the hemoglobin percentage? That would be something like 97 percentage. 97 percentage. So this is 97 percentage. When the partial pressure of oxygen is 95 torr, the percentage of hemoglobin saturated with oxygen is 97 percentage. But the same blood, when it comes into the after coming into the cells, the partial pressure of oxygen will become 40. Okay, partial pressure of oxygen in pure blood is 95. Partial pressure of oxygen in impure blood is 40. When it is 40, what is the hemoglobin percentage? This is the this is here 95. It is 97 percentage, and at 40 it is how much? So let us see. That would be about 75 percentage. It will be 75 percentage. So here it is 75 percentage. So what's happening here? The percentage of hemoglobin saturated with oxygen in the oxygenated blood is 97 percentage, but that in the impure blood is only 75 percentage. So what is the difference? 97 percentage minus 75 percentage, that's 22 percentage. That means out of 97 percentage, only 22 percentage of oxygen has been delivered to the tissues. 22% of oxygen. That means only about 1/5 of oxygen is delivered to the tissues. What about the remaining 4/5 of oxygen? That is retained with the hemoglobin. That is present in the hemoglobin. All oxygen is not given to the tissues. Okay, here 97 percentage is the hemoglobin percentage. Here it is 75 percentage. That means it has delivered. That's called as unloading. Only 22 percentage of oxygen to the tissues, and only 1/5. 22% is approximately about 1/5, and the remaining 4/5 of oxygen it will keep it with itself and provide that oxygen in emergency conditions, like during doing vigorous exercise or when there's more demand for oxygen, like that. That's why even after a person is dead, the hemoglobin oxygen is still supplied to the tissues up to 4 to 5 minutes. After a person is dead, the oxygen from the hemoglobin is still supplied to the tissues for about 4 to 5 minutes. That's because of the remaining 4/5 of the oxygen. So this 4/5 of oxygen is always kept with the blood. It's about 70-75 percentage is always kept with the blood, and that is in reserve. The oxygen is in reserve, and it will supply that oxygen when there is an emergency condition, like when there is a lot of work to be done, when a lot of energy is required, or when a person is dead. I mean, in emergency conditions, this hemoglobin with the remaining 4/5 of oxygen will give the oxygen to the tissues. So that's it. I hope it was helpful, and thank you.